when its wavelength is lesser, so and its energy is very high and it will decompose very faster. So we are going to use so many lights indoor, and natural light is also present. So our product are going to be getting degraded by these light sources. These reactions can be induced by the exposure to the photolytic sources emitting in the range of 290 to 800 nanometer region. So this is highly damaging. So which are prevailing in our regular environment also. So less than this and which are existing, so it is specific purpose in the equipment what we are going to use for the testing, but we are going to use the more than this. It is present even in the IR waves and radio waves, everything is existing. It is not going to have a much damage when compared to lower wavelength. So the relation between the wavelength and the energy of the various form of the light, if you see it, then you will be able to come to know that how to control my light so that I will be having a more stability of product maintained. The first thing is UV light, that is having a wavelength from 50 to 400. Generally we use in our pharmacy 200 to 400 wavelength in our equipment. And visible light, it is 400 to 750 in the VIBGR region, that is what we have got in rainbow. So this color range which is present in this region, and IR region, that is infrared region, 750 to 10,000. So the higher the wave number or lower the wavelength, so we are going to have high energy kilocalories per mole. So that is going to give the impact of more onto our product. So it is going to have a high degradation potential. So the photolysis which can be prevented by the suitable packaging in the abundant color bottles, we generally suggest them, and the cardboard outers, so that light will be present as a not transparent to those cardboards, so it will not be present inside the packaging. And aluminum foils and vapors, they also provide the dark atmosphere to avoid this particular one. And we are going to see the pathways and how we need to have, and how it is going to be affected in detail little bit later. So this is 2 marks question. Whatever it is present we are attempting now, then we will go to 5 marks, and then we will go to 10 marks question in the order what you are going to be have this session. And the second one is moisture. So it enhances the hydrolytic degradation. We know that generally it is assisted by the presence of either acidic or alkaline environment for its more progress. And packaging materials such as the glass, plastic are usually chosen to prevent the exposure of the drug product to the high humid conditions, because they provide closed atmosphere and it will prevent the transit of moisture in and out. This is actually what is required in a closed container. And microbial instability. Most of the times we are getting entered with these microorganisms from different sources. The major source which we are going to have in our formulation, which is going to impact our stability, is water. We know that water which is present in our pharmaceutical industry, there are 4 types of the water. What is the simple raw water, which is present from the well, or it may be taken from the municipal resources. So the raw water which will be used for the gardening purpose, the municipal resources are purified water which will be used for human consumption in the cafeteria and washing of our equipment we are going to use. And then we are going to have water for injection, that is used for the injections, and sterile water for injection, that is required to be filled in the ampules to be broken to make up the reconstitute the parental dosage forms. This water, what we are using, these 4 categories which are going to enter into our product may be having a chances. So we are going to have these gram-negative groups, Pseudomonas and Xanthomonas or Flavobacterium. These are all going to enter. Of course, our pharmacopoeia provides colony forming unit limit. See if you how many colony forming is which are going to be present allowed in the particular dosage form. The second source of the microbial contamination and which will affect the stability is the air. So we are going to manufacture our product in an atmosphere where it is present, air is present. Now we cannot manufacture everything in vacuum. So this air is inevitable, and in the environment which are present, so air is going to have dust particles, and in the dust particles are the droplets which are coming from the coughing or any personnel they are entering it, they will be causing the entry of these microorganisms through the droplets or the fibers and the dust particles, and they will be causing a contamination. Why I am telling this is no organism they will be present and floating. Even that COVID virus what we are facing now cannot be flying and it will be floating or in the by themselves. They cannot do all these things by themselves alone. They need to be seated onto the dust or a droplet. Then only they will be entering and infecting it. This is the major fundamental we need to observe. So if we want to clear and purify our air, and we are able to remove all the dusts and fibers and droplets, it means we are going to remove 99% of the viral load, or it be the microbial load. So the presence of these as a substratum, on that these microorganisms will sit and getting entered into the one place to another place. So we are going to do the air filters to avoid this particular problem, to improve the stability in our workplaces. And almost all pharma industries, if you visit, it appears like a 7-star facilities, move beyond that, because we are going to have this air supply and a proper water supply, and which is having the highest utmost care, which will be taken not only the visible organisms and particulate matter, non-visible particulate matter and organisms we need to take care. So by this way, we can avoid the contamination of the microbial microbes into our product. Another major source, the third one, is the raw materials, which we are going to use as an excipient or a drug in our pharmaceutical products. They will be having some contamination, they will be entering into our product. This is actually, we can be removing it by asking the vendor, you purify it if it is present any colony forming units beyond the limit. So we need to use the ultrafiltration. We have already learnt in the engineering. So we are using the membrane filters by converting a drug product into a solution, and the solution is filtered and removal of all microorganisms. After that, we are going to recrystallize them by evaporation and making it a supersaturation and getting the crystals which are having devoid of all microorganisms, which is the approach which can be used to prevent the degradation by the microbes which are occurring by the contamination of raw materials. Another major issue which we are going to get is, even though the starches and the pigments which are having in our excipient which are prevented, and we have already avoided them by doing all these steps, but they are the good medium to encourage the growth. So what will happen, even one microorganism or the microorganisms which are present in the permitted limits, also they can multiply and cause the damage to our product. Because of that reason, we are using a preservative. That preservative will not allow these microorganisms which are already contaminated our product within the limits also, they cannot grow and they cannot show their presence in the higher number. Like this, we are avoiding the microbial instability. I don't want to take up more in this particular topic, because you are having more and more in the microbiology subject you might have already learnt. And as a part of extension of learning for the MBA purpose, we are learning this in the syllabus of stability also to aware ourselves. Another major contamination is the gums that is used as a binder, or it may be a suspending agent. We are using it. They are the good sources of the microorganisms like Actinomyces, and the animal products like what we are using, a gelatin. So these are all the products which are having a coliforms and salmonella are getting affected by these animal products. And personnel who are working in that particular factory, they can transmit these coliforms and staphylococci and streptococci. They are obviously present onto our hair and skin, and droplet infections will be present. This is the reason, if anybody is working in a pharmaceutical industry, he has got a disease or any communicable disease, they will not be allowed to enter into the product factory for the manufacture or the work. They will be kept alone, and separate facility will be present at the security at the entrance gate. If anybody is having any communicable disease, they will not be allowed to enter. This is actually what we are going to avoid, such type of problems in our workforces by the personnel. And also, somebody are not aware that he is having some problem and having a microbial load very high. So we have got a PPE, personal protective equipment that will cover entire body. Whenever we are going to our workplace, it will prevent the microbial contamination so that our stability will be enhanced. So these are all the fundamentals what we are going to use in our workforce so that we will be avoiding this contamination. And most of the times, if you are entering after completion of this B Pharmacy course and you are joining the job, the first thing what you require to do is after entering and punching your entry by biometric or finger, and you are entering and going directly to the bathroom, and where you are having a wash of your hands and face. Sometimes you will be having a facility to have a bath also in pharmaceutical industry, and leave all your street clothing and keep the side. They will be provided with lockers, and you need to keep your jewelry and watch and mobile phone, everything in the clock room, which is assigned to particular employee. And we are changing our dress of a street cloth and street supply and keeping separately in our clock room, and changing over to the company wear, where they will be providing a separate uniform and they will be having a PPE that we need to wear. This is the reason our product manufacture is very costly. Even this is the making you to not to go to the pharmaceutical industry for training. You may be thinking that, sir, you are telling and everything is appearing like something fantasy and we are imagining it. Why can't we go and see them? If it is the case, and all 448, say for example, our B Pharmacy course, we have got 400 students. If you are visiting your factory for a training or anywhere, they need to use 400 PPE equipment for your purpose, and they need to throw it out or wash it out. It is big expense to the company. That is the reason they will not allow you to go to the particular premises, so that it is very costly for them. And also not that we are going to have little bit indisciplined behavior if it is present because of unawareness. So that will cause the damage to some of the equipment or the infrastructure. So this is the problem. We are devoid of this, but we are proving that we are in a project or somewhere we are going to have an opportunity to see them. Of course, we have got a online content. They will be available virtually and suited, already pre-suited or photographed videos are available to see them. So if you Google them and the pharmaceutical facilities of injections or anything in Google and YouTube, you will get a lot of things. You will learn that as this much care will be taking to prevent the microbial contamination. This is actually what the criticality of it. I would like to enlighten little bit more what the impact is. If anybody is present in the production system, if he is an in charge and he will be working and handling all the in charge of the operations which are present there in this factory or premises where he is working as an in charge, if it is found in an insect leg or excreta or it may be vermin, anything which is present in the premises, it means that the person is going to lose the job. Reason is that if we are not able to avoid the entry of living microorganisms, they are visible to the naked eye, and how can we avoid the entry of microorganisms? Completely, they will be making the strict action because of that capability of the particular head. They will be making the critical actions, because otherwise that will be entering into the product. Even if somebody is finding a nail or a hair piece or insect leg onto the dosage form, and if you are working as a drug inspector after completion of the B Pharmacy course, you can ask the company to close. How can you expect a tablet or a syrup or is going to contain these debris and it is debris which is going to cause the damage which you are not supposed to keep, because we are not claiming that in my label I am giving the nails or insect leg or insect excreta. When you are not claiming that, how can you give to the patient when you are charging and paying the money? This is actually outstandingly having a regulator mechanisms where it is preventing, even though it is invisible, we don't have any option to prevent them. We need to take utmost care in our building design, equipment, entry of facilities and wiring and electricity and supplies of anything which you are going to have a design that will not encourage the growth of these microorganisms, and they will be prevented, and they are entering also, they will not be having a survival inside them, because we have got a disinfections and we have got a sterility stages where which are going to have a class 100 zones. We have got so many interlocks and we have got multiple door entries, and These are all going to prevent, in addition to your air curtains and false barriers, everything it is going to have. I think you might have already seen the laminar workbench in your laboratory of microbiology. Even some microorganism is entering onto your product in the laminar workbench that will not allow you to the microorganism to enter directly falling onto your media. Even though it is present in your hand, it will be taken to the exhaust. Such facilities which we are creating so that we are having a benefit of not having instability because of this microbial contamination. The second thing which is having a major impact onto our product is packaging and impact onto the stability. So we are got a major packaging material is a glass. So most of the times it is a glass is resistant to the chemical and the physical change. It is the most commonly used material. So which we are using. So, but the limitation of it is having an alkaline surface, so it is more prone to have a hydrolysis if your product is containing water. So that is the reason we are going to use a borosilicate glass. It is not going to have alkaline leachable in that. So we can overcome it. The slide whatever you are seeing is a GPAT questions. So how to overcome the problem of glass to enhance the stability of your product. The second thing is ions may be precipitate into the insoluble crystals from the glass, which are present already in the glass, alkaline earth metals, or it may be having a divalent cations and anions which are present, they may be getting precipitated. For that purpose, we are going to use a buffers to avoid the change in the pH of the system so that we will not be having a alkaline or acidic hydrolysis. Permits the transmission of the light, which may be accelerate the decomposition because a transparent container of a glass, it can be allowing the light to enter into the product. So we need to use an amber colored glass that will prevent the entry of UV light into the product. So we can safeguard the stability. This is what we are going to use it as a minimum consideration whenever we are packing and labeling our product. We need to take these issues into the account when we are developing a product in initial stages itself. We need to concern this, otherwise we are going to fail our stability because we are studying initially in the poly bags or somewhere which you are having highly tight containers and metallic containers. Later onwards you are launching your product in glass. These issues will come. I hope you understand the gravity of it. The second major material which we are using in our pharmaceutical industry is a plastic. We have already learnt in the material of plant construction in the previous pharmaceutical engineering. We have got a plastic, polyethylene and polypropylene and nylon. So these plastic material with high density polyethylene, HDPE and LDPE, low density polyethylene, depending upon purpose, we are going to use them because of their flexibility, and they have got continuity and without having any openness, and they will be having. But the problem with the plastic is migration of a drug through the plastic into the environment. Plastic is a flexible membrane like structure. So the drug will cross and entering and leaching into the packaging material and reduce its concentration and cause the damage to the patient because the concentration is going to be less when it is entering into the package. This is very much crucial in the case of preservative and the colors and dyes. They will be adhered onto your surface and the dosage form will become faded, or the preservative will be taken and absorbed by the plastic, and you will get microbial contamination into the packaging. So these things which we are going to account when you are testing our stability in advance, so that we will not be having such problem by keeping them and studying it in the laboratory, that any reduction in the preservative or a drug or a dye is present when I am keeping my product inside these containers. Transfer of environment, moisture and oxygen and other elements like your contamination with another product of adjacent factory, or it may be a dust, anything into the pharmaceutical product, that will be avoided. So if you are having a screwed container, we need to have a small gasket or a plastic structure or rubber structure so that it will be having a leak proof and a closed container. Providing a closed container. Leaching of the container ingredient into the product, because we need to have a specific flexibility. We want to have a specific color, because If we want to give a child dosage form, we need to have a peculiar color, and we want to give a different shapes to enhance its aesthetics. That is the reason we are going to give a plastic container a different shapes. For that purpose, we are using colors and plasticizers. They will be leached. They are highly carcinogenic in nature. So they will be entering into our product. People are consuming, they will get cancer. So we need to avoid in advance in those cases whenever we are using these container in our dosage forms. Whether it is any product which is going to have entering into our body, we need to test the presence of leachables and study the toxicology. So in the toxicology studies, we are going to read this carcinogenicity, that is cancer causing capability of the leachable. Otherwise, the patient will get cancer in due course of time if anything which is going to have a carcinogen entering into the product and he is taking and consuming it. Not only that, nowadays the regulatory authorities, they are insisting teratogenicity. Teratogenicity means the person who is consuming may not have any effect, but his offsprings, his or her daughter and granddaughter may get affected with that. That is actually called as a teratogenetic effect. In the teratogenetic effect, the patient is safe, but the next generation will be getting affected with the problem. So we need to study the toxicology of the material which you are allowing it, whether it is a cleaning agent, or it may be a dye, or it may be a plasticizer, or anything which is coming, entering into as a impurity. So what is its effect? And we need to avoid that if it is present, it is proven. And adsorption of the active ingredient or excipient that by the plastic is a major issue we need to study before we are selecting this plastic. In every company in B Pharmacy after completion, you can apply for packaging development department. It is very good and highly elated, and you will be feeling like you are doing a design. So you may be having a fashion design or something. There your work is how to make your appeal, your product in the packaging good to the patient, how we are making the regulatory authority happy by not having these issues. They are all being studied by the packaging development department. So you yourself have got an ample opportunity to work in them. So you can apply here, and that is also a very promising area where you can design your product for the lactating mother. It may be a pregnant woman, or it may be a pediatric patient, or it may be a geriatric patient, or an old aged patient. If they are holding a glass bottle and they have got a shivering, if it is falling onto the floor, it will get broken. So we are avoiding that by using this packaging specifically. Even if it is falling also, nothing will happen to the package. So the costly medicine will be safeguarded. These are all the things which we need to do as a research and suggest for the manufacturer to enter into the market. I hope you understand the gravity why we are concerning all these and sitting in the classroom and learning, so that we are going to apply even one sentence which is being taught or which is being learnt now, which will be not going to be wasted. So you are going to use somewhere. The third thing which we are not preferably, we don't use it, is a metal because of their costly in nature. But sometimes we need to use them without any skip. Such materials like aluminum and alloys we are going to use. They are containers for the emulsions and ointments and creams. There is no other option we need to use. And the limitation are they cause the corrosion. That is the reason they will be getting converted into inactive metal to active ions and getting corroded and precipitated into the drug product. One thing is they cause the damage of the precipitation, and they take the changes in the pH and causes the hydrolytic problems. So we are going to use sometimes chelating agents or complexing agents to remove those ions, metallic ions from the packaging, to scavenging them. We call it as a metallic ion scavengers. They scavenge it and they hold them not to getting affected our product. So another way is we are going to avoid these problems to prevent the contamination and degradation and enhance the stability. Is coating of the tubes with the polymers may reduce these tendencies. So internally we are coating with a plastic or a glass or something else, so that we will be having a protection that will be avoiding this problem of corrosion. So this is actually what we are going to take a decision whenever you are selecting a packaging. And the last one is the rubber, which we are generally using in the injections, where this is having a rubber cap. Through that, needle is getting pierced because it is a self-sealing. When you are removing the needle, it will completely close, it will not allow the microorganisms. Whereas if you use the piercing into the plastic or anywhere, the hole will be appearing forever, and you are allowing the microbes to enter into the dosage form. So wherever we have got a multiple dose which we are required to take, in that particular case we need to use a rubber cap. There is no other option. So there is a problem of extraction of the drug ingredients and leaching of the rubber components into the product. Both will be present. It means it can absorb some of the product which is present in the dosage form, and it will leach some of its component into the dosage. Both are problematic. So you may be having, when we are playing in the childhood, if you keep your rubber caps or anything into the kerosene or a petrol, it will swell by its 3 to 4 times, very bigger in nature. You can find that as my rubber cap has got very bigger. Reason is that it will absorb all those solvents. It means that it can absorb our medicament also. It can absorb our solvents also in the present. So it will be getting swollen, or it will be getting taken away our medicament and cause the damage. So the pre-treatment of a rubber vial cap stoppers and the closures with the water and the steam reduces the potential of the leaching. That will reduce the damage. We are going to have the less impact onto our stability. This we want to do it to avoid the further problems. Now, we have seen the major out of our syllabus, which we have got an environment and microbes. With a little bit fastness we have entered and we have discussed it. Now we are going to enter into the different types of the degradation, and we are going to see one by one and entering into our regular curriculum with the depth, so that we will be able to answer 5 marks question and 10 marks question and GPAT. Ultimately, at the end, we are going to see how to calculate the expiry date. So with the calculations which you are going to do, the same thing after completion of in the theory, we are going to have in the practical also, and we are going to calculate by your own hand and report the result of expiry date of solution which I am going to give you, and you can predict what is its expiry date. So this journey will be, obviously it is present in the practical also. So now we will be entering what are the different types of the degradation. One is chemical, another one is physical, another one is the types of degradation is the biological, based on what we have already seen fundamentally. So the similar thing is rotated and the another side of the coin which you are going to enter. So the chemical degradation is hydrolysis. It is splitting up by the water, in the presence of the water, into the 2 ions. And the second chemical degradation is the oxidation. It is inorganic and organic compounds with the loss of electron or addition of oxygen. So it will be either loss of electron or loss of hydrogen or addition of oxygen into the product. Whether it is inorganic product or organic product, we call it as an oxidation. So in those cases, we are going to avoid the exposure of oxygen into the bottle above the meniscus. Most of the times we have got a free spaces, whether it is wide or above the dosage form, we have got a space. In that space, if oxygen is present, it is bound to be entered into the oxygen. So sometimes what we'll do, we will replace that with the oxygen with the nitrogen. So we remove all air with nitrogen, so that we will be having nitrogen atmosphere and blanketing effect. So we will be avoiding the problem. How will you know? If you know this in advance, if the presence of the oxygen is going to cause the damage, so what you require to do, you suggest your packaging people that you create a atmosphere nitrogen in the bottle, so that you will be enhancing your stability by avoiding the oxidation reactions. So I think you may be seeing some of the Lays or chips packets. What you are observing, it is just like a fluffy, and they are selling only the air in the name of your product. So they are very fluffy in nature. It is filled with only nitrogen. Now product is very less, very less there. If you open the bag, it will like just a pillow. It will appear, but it will contain oddly 5 to 10 chips only. It is costing around 10 rupees at the cost of maybe 1 rupee 1 chip. It is that much costly, but we feel that it is they are giving too much. So this is actually what they are doing, is filling the air there. So so making it fluffy and selling it. So, but the problem in this particular case, in the case of coffee powders, or if you're taking a Nestle bag or any bag where you're taking it, it is containing bags in a pouches which is fluffy in nature. To retain the coffee flavor, we are going to use it. It means even though you are learning the concept now in the pharma, which you can apply for the food industry also, some another way also, the similar concepts. And you feel like whatever the object you are seeing outside as a product onto your hand, you will think that yes, this is the entire journey might have undergone in this particular packaging and making it appearing onto my hand. You feel like as contented and satisfied that as I have got enough knowledge after seeing that package, this much that crop is present. Of course, we have already seen the chemical stability is the photolysis and another form of a chemical stability which is caused by the temperature. These are all the things we will be seeing one by one. Now, you will be asked the question, what are the different types of chemical instability which is existing? List them. Just you need to write, one is the hydrolysis, second is the oxidation, third one is photolysis, another one is the temperature that will cause the chemical instability. But you will be asked the question, write the different parameters and the, how to prevent them with respect to oxidation or any particular one, 5 marks question. You need to write it separately. It is little bit more. So we'll be seeing a 5 marks question it is been having. So you need to write some answer, and if you are 10 marks, you need to elaborate further. In the similar line, we are going to enter. So our answering, because these questions we are going to get it in the term and also sessional also. We are going to have in practical also. Not only that, it is very important for our survival in the business of pharma. Some of you may be becoming entrepreneurs. So this is the last stage of your business where you are going to enter into the market and make the party to the teammates. So the chemical degradation of the dosage form occurs through the several pathways like hydrolysis, oxygen, and because of that it is having a decarboxylation or photolysis or racemization. So what is this racemization whenever you are having it by the impact of these conditions? So most of the times some of the forms, now you may be having 2DG which is being evaluated, now it is going on in Delhi, evaluation is going on. Dextrorotatory glucose, deoxy, D-glucose only, not L-glucose. It means we cannot use the racemized product of a glucose. L and D form we cannot use. Only D form can only act. Because most of the times we are taking L-dopa. We are taking levorotatory, not the dextrorotatory, L-dopa. It means our products are going to contain both dextrorotatory and levorotatory. We are going to use one form. So if that one form is converting into another form, your efficacy will be reduced by 50%. It means you are starting with D and your end product is destabilized to D and L. So D is converting into a L means 50% D will be present, D is 50% L will be present. If D form is active, L form is not active, it means your stability is causing a damage to your product. Only 50% of given dose will be working. Remaining 50% will be not active, even though you are paying for it and it is not working. So patient disease will not be getting cured. This is actually a biggest problem with the chemical degradation and converting into a racemized mixture. Most of the times our body are aligned to some it is a nature itself, it is aligned like that. One receptor is present. So we are going to have a opposite get attracted. In that particular case, levo or dextro is specified for the action. So in those cases, we are having a naturally available in DL form. In the racemized mixture, we need to separate and we need to keep them, it is stable. By maintaining the stability, by maintaining the pH, like that, we are going to have a particular form. So which may lead to the lowering of a therapeutic agent in the dosage form and the formation of a toxic product and decreasing the bioavailability. Now you may get the question, sometimes your racemization or a degradation product may not be causing a damage. Sometimes it is more toxic than your actual poison. So that may kill the patient or cause the damage by causing a reaction, or it may be rashes or any rise in the temperature, or it may be a pain in particular part or numbness in particular area. So these are all the toxic effects which are going to have this. Second one, we have already seen that in this particular chemical degradation, the major effect what we have seen is the hydrolysis. It is a important in the system containing the water, and we need to consider with utmost care in case of emulsion, where we are going to use water and oil also. So water is obviously present. Suspension, obviously we need to have water as a major. Another one is a solution. So these are all the things bound to contain a water. If your product is containing any ester or amide, it is getting hydrolyzed to free carboxylic acid or amine or alcohol. So it is not having activity. The product will not have activity. So because of that reason, we need to avoid it, especially where you have got a problem of presence of water in the dosage form. Say for example, you have got a tablet. Your tablet is made up of a solid which is not containing any water. Then hydrolysis degradation in the tablet is much lesser than your syrup or a solution or a suspension or emulsion. This is what we need to understand. It means if your drug is having hydrolytic potential, if you want to give them, then what you require to do is deliver them as a solid dosage form instead of giving as a liquid dosage form for enhancement of its stability. Now you may get the question, sir, for the pediatric patient and geriatric patient, like old aged patient and kids, they are having problem of consumption of this tablet and capsule. They get choked with the throat, within the throat, this dosage form will get choked. Nobody will prefer. In those cases, how can you do it? So you may be having a pattern in the medical shop. Whenever you are bringing, they are asking you to take the bottle and boil the water and cool the water and make up the volume of the mask, shake the bottle and consume it within 7 days. That means you are giving as a dry format. Why it is been introduced like that into the market? Why you have suggested as a pharmacist to release into the market? Because in the stability, you already estimated that this is having a problem of hydrolysis. If I am keeping the product like that, it will degrade within 2 months. So it will degrade to the less than 90% within 2 months. So how can I launch this product within 2 months? It will not be sold. It requires minimum 2 years. Whereas you are dispensing the same product as your dry product, then it will be stable for 5 years. So after making up with the water, it will be stable for 2 months. So you are going to consume within a week, because every day you need to take 5 ml. So morning and evening 5 ml, 15 ml is over. Around it is given for the 4 days. So around 60 ml, or it is 7 days, it is 75 ml will be there. So you are going to have the bottle which will be consumed. Later on you are going to throw. So this is actually what you have got a double expiry dates. In the stability testing, we need to understand the concept of double expiry date. What is the double expiry date? In the stability testing, you are going to see first, before reconstitution, what is the expiry date of the bottle without opening the bottle. That is having higher expiry date like 5 years, 4 years or 2 years like that. After opening the bottle and making up and reconstitution, what is its expiry date? Both you need to study in advance. And you need to prescribe there in the bottle label itself. After reconstitution, consume it within week, and if you don't consume, throw it. Don't use it, throw it. And you may, most of the person, they may be thinking that I have purchased the product and I'm paying the money. So why should I throw? I drink it. No need to drink. It means you are drinking poison. Why do you want to drinking poison? Even though it is getting wasted, let it be wasted. Put it out. So this is where we need to enlighten the society as a pharmacist. If we understand this concept, we know that even though it is causing a wastage, it is not worth to drink that, because even if you drink that, it is going to have more damage than the benefit. Why do you want to invite damage to your body? So we need to throw it. Why you want to use it? So in the double expiry date, it will alert the patient so that after opening and before opening, you have got 2 expiry dates, you follow it. Then only this will work. And these are all insert and instructions you are giving on the label based on these stability studies. How you have come out of this idea, where you are giving, you don't want to give as the prepared suspension. You want to make a dry suspension and making the patient to reconstitute it. Why you want to do it? You found that in the stability studies, you have found that this is getting degraded very fast. That is the reason you are doing. And the other way, you may be having your eye drops and ear drops, it is having a 2 expiry dates. So there, what you are doing, first, before opening the packet, it may be having 2 years. But after opening and use it, within 1 month, you need to consume it. Otherwise, what will happen? That will cause the microbial contamination and you will get eye infection. You want to treat your eye with a problem of a disease, but you are giving an infection to the eye by using longer periods. It means you have opened 1 year back one eye drop. It is better to throw, not to use it. Otherwise, it will already got contaminated with your tips are getting contaminated, and it is getting contaminated and cause the damage to the eye by using them. That is the reason instruction what you are going to do give there. While testing your stability, you found that don't touch the tip of the dropper onto your surface of the body. You keep it, don't touch it. That is one. And don't touch your tip with the fingers. This is what you are giving an instruction. The third instruction what you give, if you find any problem like a particulate matter, throw it and consult your physician. That is another one. Because it has got precipitated or dust, we should not allow your eye get damaged. And the fourth instruction what you are going to give generally is, if you are finding any irritation and or any side effect, you stop using it and meet your physician. It means that the drug is not suitable to that particular patient, so you should not use it repeatedly. These are all the things which we need to have an enlightenment, so that we will have a suggestion to give to the patients. Most of the times we are having another zone in the hospital where you can get a job is the drug information services. In the drug information services, you are going to give information to nurses. You are going to give information to patient, lab technicians, and parents are the attendants of the patient, not only them, and doctors. You are going to give the instruction. When he is prescribing wrong, or he is giving he need to be given information that the recent advancement, it is suggested that it is a contraindicated, and both the drugs you are not supposed to give. It will cause a damage by giving a drug interaction. So this is all the problem which you are going to have the stability issues after when you are using it with another product. Another product, when you are using it, it will be going to cause. So these are all the things which we are going to study as a part of our stability program. And most of the times we are going to use them simultaneously with another product during due course of time. We are going to give as a modification of our insect and writing them in the contraindication. So this hydrolysis also, the drugs which are affected by the moisture and water vapor from the atmosphere, and usually categorized by the hydrogen ion and hydroxyl ions. So we need to maintain the pH in the stable near to the neutral by adding a buffer. So this is the way we are going to control. And in this active drug is decomposed with the solvent. That is the reason sometimes we remove the water, add oil and some another solvent, non-aqueous solvent. You may be finding these oily suspensions in case of amoxicillin. Some of the cases you may be having where reconstitution is not required, but their taste is not good. They will be having a bland taste. You don't prefer them, but you need not do any reconstitution because you are outside. You don't have any facility of heating water and adding that. So in that case, you need to purchase the non-aqueous syrup only by avoiding the solvent. Usually, the solvent is water. Sometimes reaction may involve the pharmaceutical co-solvents such as ethyl alcohol and polyethylene glycol. Such things which you can avoid. Sometimes you need to add to reduce the potential of hydrolysis. Both are required to do. Sometimes you need to avoid, sometimes you need to add co-solvent. Main classes of the drug that undergo the hydrolysis, generally you need to bother is esters and amines. And most of the times in the presence of alkali and acid. So whenever you are handling these drugs, you do take care that should I add the buffer, should I add the avoiding any co-solvent, certain things which you need to understand. Most of the times if you see the ester hydrolysis which are involving the breakage of your ester bond, like you have got RCOOR, This is actually what your so many drugs are present. Of course, this is R1 and R2. Both cannot be there. In some cases, it may be the same. In that particular case, the R is a functional sum of the drug which are present with a carboxylic group in presence of the water H2O. So it is present with enlightened, that is catalyzed in the presence of acidic or alkali, converting into carboxylic acid COOH and alcohol OH. So this is actually what is generally it is present in the hydrolysis reaction of a ester. There is the amide which is also present. So it is more stable than the ester. Preferably, it will be used in case of esters to enhance the solubility in the lipid. We are going to use this tendency to convert into a ester or amide, because we know that the carboxylic acid, they will not be getting amines pure. They are polar in nature. They will not have the bioavailability. To enhance that, we are preparing these salts. So these conversions which you are going to use it, wherever it is having the stability is there, if you want to convert, it is better to make instead of ester, it is better to make amide, because they are more having, this is the GPAT question. So you will be asked the question in the GPAT. So which is more stable when compared to your ester or amide? Amide is more stable than the ester. But susceptible to the specific hydrolysis, acid-base hydrolysis, most of the times, which is having, it involves cleavage of the amide linkage to give an amine instead of alcohol and ester. That is in the case of ester, and you are going to have a carboxylic acid. The example is you have got RCOONHR. So this is cleaved into RCOOH carboxylic acid and RNH2, that is amine. So some of the examples of the amine drugs are chloramphenicol and barbiturates. They are used for the hypnotic we are using, and you might have already studied in the pharmacology and the medicinal chemistry. And whereas the ester drugs, preferably we are using is atropine and aspirin, physostigmine and procaine. So adrenaline, these are all going to be used. Some of the functional groups subject to the hydrolysis, and they're having some more things which are being capitulated as a